In this video, we will be seeing about the biosynthesis of heme and its related disorders. Heme, or what is referred to also as heme protein, has several important biological functions in the human body. So as we see the importance of and role of heme, it is responsible for oxygen binding and transporting that oxygen to the different parts of the body. It participates in electron transfer. It is helping as a catalyst in various biochemical reactions. It is participating in signal transduction and is also participating in micro RNA processing. The organic compound heme contains an ion atom which is in the center of the porphyrin ring structure. You can see it on the slide. The two major organs where this heme is produced, or the biosynthetic pathway takes place, is the bone marrow and also the liver. In both, in the mitochondrion as well as in the cytosol, part by part, these pyrrole ring structures are synthesized, or the porphyrin molecule is synthesized, and this synthesis involves various enzymatic reactions. So like mentioned before, the heme synthesis partly occurs in the cytosol and partly in the mitochondria, and it involves a total of 8 different enzymatic steps. The beginning of the reaction is with the condensation reaction between succinyl-CoA and the amino acid glycine. You can see the reaction over here, which is catalyzed by ALA synthase enzyme, and the end product what is got is delta amino levulinic acid. This delta amino levulinic acid formation also requires the presence of pyridoxal phosphate. Further, this delta amino levulinic acid which is formed is then transported from the mitochondria into the cytosol, and in the cytosol, two molecules of ALA, that is delta amino levulinic acid, combine, condense, and they form the first pyrrole ring structure, which is called as porphobilinogen. So you see over here the formation of porphobilinogen, and the enzyme involved is alpha amino levulinic acid dehydratase enzyme. In the similar manner, further 4 molecules of porphobilinogen form, and then they condense or combine with each other and form the intermediate hydroxymethyl bilane. So here you see the structure of hydroxymethyl bilane, and then this hydroxymethyl bilane, which is also short formed as HMB, it undergoes a ring closure to form the pyrrole ring D structure, and this ring D structure is called as uroporphyrinogen 3. This uroporphyrinogen 3 formation is catalyzed by the uroporphyrinogen 3 synthase enzyme. The side chains of this uroporphyrinogen 3 then undergo different types of modification to first form coproporphyrinogen 3. This coproporphyrinogen 3 then gets transported again to the mitochondria. It undergoes decarboxylation reaction in the presence of an oxidase enzyme, and it forms what is called as protoporphyrinogen 9, which you can see on the slide, protoporphyrinogen 9. Finally, this protoporphyrinogen 9 then uses the oxidase enzyme to form protoporphyrin 9, and then protoporphyrin 9 gets inserted with the ferrous atom or the ferrous ion by the ferrochelatase enzyme, and then we actually get what is called as heme, which is going to combine with the protein globin to form hemoglobin to carry oxygenated and deoxygenated blood back and forth from the heart to the different parts of the body. This ferrous protoporphyrin 9 which is formed has 4 pyrrole ring structures with a methylene bridge which is connecting all of them, and this heme has variety of biological functions like I mentioned before: oxygen transport, It is a prosthetic group in all the cytochrome P450 enzymes. It participates in the electron shuttle in the ETC. It is responsible for cellular respiration and also in the regulation of circadian rhythm. Thus, we see that this heme formation is a very important process that is taking place, and it also needs what we call as regulation. So there is regulation of heme synthesis, and these are the five major sites where heme regulation or heme synthesis regulation takes place. The first site is that of the ALA synthase enzyme. Then again there is ALA synthase, which is existing in its allosteric form, and it is inhibited by the hematin formation. So whenever hematin concentrations increase, ALA synthase enzyme will be inhibited from its action. Similarly, when an individual suffers from deficiency of vitamin B6 or is exposed to heavy metals like lead, in these cases also there is going to be stoppage of heme formation. So that is why you will always see that a person who is anemic in condition will always be deficient with vitamin B6. So the treatment involves increasing the RBC count by simultaneously supplying vitamin B6 also to the individual. Similarly, concentrations of erythropoietin also regulate heme synthesis. Now, this deficiency or absence of these enzymes is going to also cause or lead to various disorders or diseases. The main disorder is what is called as porphyria. So porphyria basically means like it is a group of metabolic disorders which is related to the accumulation of the porphyrin, which is happening mainly because of deficiency of one or more than one enzymes which are responsible for heme synthesis. And then depending upon its intensity, different types of symptoms or damages are seen in an individual. At a very low rate porphyria, there will be only simple skin damage, or there will be photosensitivity, but or there can be change in urine color. But when excessive amount of porphyria is present, then it can lead to some severe disorders as well. So this is how it can lead to photosensitivity or skin damage or different neuropsychiatric disorders because of porphyria, or these are the major signs and symptoms of porphyria. Porphyrias basically can be classified into two types: the erythropoietic porphyria and the hepatic porphyria, and both of them are occurring either due to a genetic disorder, or it is due to a liver dysfunction, or exposure to heavy metals like lead, like I mentioned before. This is the classification of porphyria: acute intermittent porphyria, variegate porphyria, hereditary coporphyria, porphyria cutanea tarda, erythropoietic porphyria, and acquired porphyria. Thank you.